talked about a lot of different metabolic pathways. Some of them were heterotrophic, some of them were autotrophic, including both chemoautotrophic and photosynthetic. And then photosynthesis and chemosynthesis, of course, are usually intimately tied to carbon fixation. And so there are many different pathways of carbon fixation. We're not going to have time to cover all of these. Most of you have learned about the Calvin-Benson cycle before, presumably when you learned about green plant photosynthesis. But microbes, as with many of these pathways I mentioned last time, were more diverse, right? So microbes have evolved many different ways to kind of make a living, so to speak, metabolically speaking. So, and some of them do use Calvin cycles. So cyanobacteria, maybe not surprisingly, use that. And of course, their, sort of descendants in green plants use that. There are some chemolithoautotrophs that use Calvin cycle, but there are others that use something called the reverse citric acid cycle. So when you learned about metabolism, you learned about the Krebs cycle or the so-called TCA cycle, right? So it turns out that microbes, some of them can run this cycle in reverse. So instead of breaking down glucose to form CO2, they take CO2 and fix it into glucose or other complex carbon. So these are green sulfur phototrophs that do this, as well as some of the thermophilic hemolithotrophs. There's another cycle called the hydroxypropionate cycle, which is used by some of those green non-sulfur bacteria like the chloroflexus that I showed you a picture of last time. You don't have to memorize all of these, but I just want us to get a general picture or a reminder of what carbon fixation pathways are all about, right? And up here at the top, you remember you're almost always bringing things in from the light reactions if you're a phototroph, or from the chemical energy developing reactions if you're a chemotroph. And so what we're looking at are those reducing equivalents, things like NADH or NADPH, FADH, as well as ATP, right? So those come in and serve as a way of providing the energy and the electrons needed to convert CO2 into reduced carbon, right? And so if you look at the overall stoichiometry, it requires a lot of ATP, a lot of reducing equivalents. Mix it together with some carbon dioxide, and out the other end, you get sugars, right? Okay. And you regenerate your intermediates, right? The oxidized forms of these molecules. So this is no different than what you learned about with plant photosynthesis in general biology or in other advanced biology courses. The reverse TCA cycle is essentially very similar to what you learned with the Krebs cycle, as I mentioned earlier. But because we're running it backwards, we're going to be using citrate as an intermediate to then create acetyl-CoA, which, if you remember your metabolism, particularly if you're a micro major and you're taking bacterial physiology, acetyl-CoA is that real central intermediate in terms of generating carbon that can then go into different pathways. It's converted to pyruvate by the addition of a carbon dioxide. And so essentially what happens here is you generate a 2-carbon molecule, turn it into a 3-carbon molecule, triose, okay? And then you can couple together 2 3-carbon trioses into a hexose, right? So in the end here, the stoichiometry is a little different, but the whole point is the same, and that's what you want to focus on. Many of the enzymes you may have had to memorize in other classes are used here, but they have evolved to be running in the opposite direction, okay? And it's possible that this is one of the earliest autotrophic pathways. It is likely that Calvin-Benson was not the first one that evolved. So something like a reverse TCA cycle may have come first, or the hydroxypropionate cycle may have come first, okay? This is only found in Chloroflexus type organisms, which is one of the deepest branching photoautotrophic lineages. As you may remember from last time, they also can function photoheterotrophically, but we don't know what they evolved first. They may have been autotrophs first and then developed the heterotrophy later. And so this is another candidate for this early ancient carbon fixation. I'm not going to go through all of this, but again, we have intermediates like acetyl-CoA. You're sticking carbons on. You have to input ATP to make that happen. Okay. And in this case, you end up with glyoxylate, a 2-carbon intermediate, which can then be used to couple together to make sugars or other cell material. So I just want you to understand that there are more than one pathway of carbon fixation that are used by different groups, particularly these chemolithoautotrophs and the non-oxygenic phototrophs use these different pathways. You may remember that when we read the paper about the desulfurudus audaxviator, that there were genes found in that genome, which suggested that it may be autotrophic using some of these alternative pathways too. So that's just another example. Okay. Any questions about that? So that is a very rapid overview of metabolism. You don't have to memorize